Welcome to The Explainer. So today we're going to unpack something pretty incredible. The fascinating medical mysteries hidden within a single, seemingly simple drop of blood. It's something we all have, something we all occasionally give up at the doctor's office, but we rarely stop to think about the absolute universe of information swimming around in those little vials. Well, we're going to change that right now.
Here's our roadmap for today. We'll start by decoding your blood draw, then compare serum versus plasma, dig into the cellular and metabolic panels, and finally, look at a really fascinating bovine blood anomaly.
All right, Section 1, decoding your blood draw. So we've all been there, right? You're sitting on that crinkly paper of an exam table, staring at a lab report that just looks like a confusing alphabet soup of acronyms and numbers. But before you even get those results, what actually happens to your blood after it leaves your arm? Well, it doesn't just go straight under a microscope. It goes to a lab for a highly specific separation process. And how it's treated in those first few literal minutes dictates exactly what secrets it's going to reveal.
Which brings us to Section 2, serum versus plasma.
This is a totally foundational step in diagnostics, understanding the distinct difference between these two vital fluids. If the lab wants serum, they literally just let the blood sit there and clot for about 30 minutes. The solid stuff separates out, and it leaves behind this clear yellowish fluid that is entirely free of clotting factors. But if they want plasma, they have to immediately mix the blood with an anticoagulant. This halts the clotting process right in its tracks, meaning those crucial coagulation factors stay suspended right there in the fluid.
Now you might be wondering, how on earth does the lab know which fluid to prepare? Well, it's all color-coded. Phlebotomists use these specific tubes with different colored tops, and each one contains specific additives. For plasma, they'll use light blue, green, or lavender-topped tubes. Those are the ones with the built-in anticoagulants. But for serum, they use red tops, or my personal favorite, the gold tiger tops. Those specifically lack anticoagulants, so the blood can just clot naturally.
Okay, so once the blood is in the correct tube, we get to the actual physical separation. After the venipuncture and either letting it clot or mixing in that anticoagulant, the tube goes straight into a refrigerated centrifuge. And guys, it is spun at an intense 1,000 to 2,000 times the force of gravity. They do this for about 10 minutes. This extreme gravity basically forces all the heavier cells down to the absolute bottom, which leaves the serum or plasma perfectly separated at the top, just waiting to be carefully pipetted into a new tube.
Moving right along to Section 3, the cellular and metabolic panels.
Now that we've sorted out the fluids, we're going to zoom in on the actual cells and the chemicals themselves. This is where those confusing acronyms finally start to make sense. Let's ground this with some insights from the Rutgers Cancer Institute. They regularly monitor something called a complete blood count, or a CBC. This is absolutely crucial in oncology because cancer treatments like chemotherapy often destroy the fast-growing blood cells that this exact test measures. So, when your healthcare team is looking at this, they're basically evaluating whether your body's essential cell factories down in your bone marrow are keeping up with systemic demand. Here's the standard CBC breakdown. First up, WBCs, or white blood cells. Think of these as your immune system's defensive infantry, constantly out on patrol for foreign pathogens. Then you have RBCs, red blood cells. These act as your vascular cargo ships, facilitating vital oxygen delivery and carbon dioxide exchange. And finally, PLT, or platelets. These are the specialized first responders that kick off the coagulation cascade the second a vascular injury happens.
14. That is a very specific and a very important number. Because while the CBC looks at the cells, the Comprehensive Metabolic Panel, or CMP, measures exactly 14 different substances in that fluid we just spun down. The CMP is basically a master snapshot of your body's overall metabolism. It rapidly checks your glucose for energy, your calcium for bone and nerve health, and 4 different electrolytes just to ensure your body's critical fluids and acid-base balance are in check. It also measures albumin and total proteins, alongside specific liver enzymes to make sure your hepatic system is functioning properly. And finally, it checks your BUN and creatinine levels to make absolutely sure your kidneys are successfully filtering out waste. All of that incredibly vital info from just 14 little chemical markers.
And that leads us to Section 4, a bovine blood anomaly.
So, understanding exactly what these cells and markers do when everything is running smoothly perfectly sets the stage for a real-world veterinary mystery. It shows us exactly what happens when just one specific blood cell totally fails to do its job. Enter bovine leukocyte adhesion deficiency, or BLAD.
First identified in the early 1980s, this is an autosomal recessive hereditary disease that strictly affects young Holstein calves. Now, calves born with this disease might actually look perfectly normal at birth, but within just a week or two, they become incredibly medically fragile. They suffer from severe, recurrent bacterial infections, pneumonia, delayed wound healing, and tragically, these affected calves usually die within just 2 to 4 months.
And this brilliantly illustrates the critical importance of those white blood cells we talked about. Remember our defensive infantry? Well, the root cause of BLAD is a mutation in the CD18 gene. This mutation causes a significantly reduced expression of beta-2 integrant adhesion molecules on the surface of the calf's white blood cells. To go back to our metaphor, the mutation literally leaves our soldier cells completely without their vehicles, those adhesion proteins, that they desperately need to actually travel to the sites of infection. So the immune system is essentially just trapped in the bloodstream, totally unable to fight off the bacteria.
So how exactly did this happen? Well, it actually traces back to a massive genetic bottleneck. All homozygous animals affected by BLAD can be traced back to a single male ancestor, a bull named Osborndale Ivanhoe. Because Ivanhoe possessed incredibly desired genetics for milk production, his lineage was heavily bred across the entire industry, and he passed on this silent mutation so effectively that at one point it was reported that up to 24% of registered artificial insemination bulls in the entire breed were carriers of this fatal flaw.
Let's take a quick look at the Mendelian genetics of how this actually operates. Because BLAD is a recessive trait, heterozygous carriers look and act completely clinically normal. They don't have any immune dysfunction themselves. But if you breed two perfectly normal-looking heterozygous carriers together, there is an exact 25% chance that they will produce a homozygous calf that is fatally affected by the disease. It was literally a genetic time bomb hidden right there in the bloodline. Thankfully, though, modern PCR DNA testing is now helping to completely eradicate this from the breeding pool.
Which brings us to our final thought. We've gone all the way from the color of a plastic tube top down to a single point mutation in a dairy cow's DNA. If a single silent mutation in one sire can devastatingly impact an entire global breed's bloodline before we even knew what to test for, well, what undiscovered stories are quietly circulating in your own veins right now? Thanks so much for joining me on this explainer. Keep asking those big questions, and I'll see you next time.